In the previous lesson, we learned about synaptic plasticity, and in combination with system consolidation, then we're starting to have a view of how memories are processed in the brain. So system consolidation involves the communication between different brain structures, the hippocampus and cerebral cortex, so as to strengthen the cortical memory trace. And when we say memory traces are strengthened, we mean synaptic plasticity. The synapses are getting stronger as a result of biological growth processes. In this lesson, we'll examine some other aspects of synaptic plasticity and then start to apply it to school settings. First, there is another kind of plasticity taking place in the brain. And much of this work was done with animals, but more and more with humans. And that is the discovery of neurogenesis in the hippocampus region. Now, neurogenesis is the creation of new neurons by cell division and takes place in only a few regions of the brain. In the hippocampus, new neurons integrate into the dentate gyrus, this region of the hippocampus. We can think of this as plasticity 2.0 because the brain is generating entirely new brain cells for future memory processing. Now, researchers hypothesize that as new neurons integrate into the already established neural networks, they interfere with prior memories. Thus, consolidating memories in the cortex would protect the memories from decay. The benefit of new neurons is that they are more plastic, that is, better able to make synaptic changes necessary for learning and memory. So if we think that memory traces are the result of neurons being allocated and then strengthening connections between those allocated neurons, if you have new neurons, inserting themselves into the synaptic pathways at sort of random positions, you're going to be disrupting the prior memory traces. So it seems then that neurogenesis is going to cause the decay of hippocampal memories. But that then would make sense of why we do system consolidation. We're going to, in a sense, transform the hippocampal memory to the cortex, so the cortex can be the long-term storage site for the memory. And this will allow the new neurons to take their role in future learning events. So let's use some diagrams to capture neurogenesis. Here we see some blue neurons, and these are the older neurons. They've already established connections in the dentate gyrus, and that connectivity pattern stores older memories. But now we have a wave of these green neurons. These are new, young neurons, and they're going to integrate into this prior neural network. Now, these new neurons interfere with old memories. As they sort of take up positions and make their synaptic connections, they are disturbing the patterns of connectivity that represent older memories. But these new green neurons are more excitable and so have a lower threshold for synaptic plasticity, and this makes them more likely to be allocated to new memories. So in a sense, neurogenesis is sort of clearing the hippocampus of older memories in preparation to make new memories. Well, 2 weeks later, the new wave of new neurons, here shown in red, integrate into the network. Again, they're going to be disrupting whatever memories were previously formed by the older neurons. The green neurons that were younger 2 weeks ago would have already been integrated into the neural network and have been allocated to memories in the previous 2 weeks. And the process, of course, then continues as new neurons are born and integrate into the network. So we want to think of ourselves as we go through our lives, we're in a sense moving through time. And all the while, we're just sort of learning new things. The hippocampus is sort of being refreshed with these new neurons. And one important consequence of the new neurons integrating into the hippocampus is that they will disrupt older memories, causing a decay of those older memory traces. But the flip side is that prepares the hippocampus for new learning. Now, neuroscientists suspect that the integration of these new neurons may actually trigger consolidation processes, so that while the integration of new neurons disrupts the older memories, prior to the loss of those older memories, the consolidation processes will save those older memories into the cortical long-term memory system. In this graph, we see the results of some animal studies that neurogenesis modulates the time course of hippocampus dependency. So here we have days after learning, and here we have hippocampus dependent memory and hippocampus independent memory. The blue line would be control animals. When an animal learns some task or learns to associate a tone with a shock or a context with a shock, one day after learning, that memory is hippocampus dependent, which means that the hippocampus is required for retrieval of the memory. 28 days later, consolidation has rendered the memory trace hippocampus independent. The animal no longer needs the hippocampus in order to retrieve the memory. But procedures that influence neurogenesis alter the time course of this shift in dependency. So X-rays destroy new neurons, and so X-rays will reduce neurogenesis, and it has an effect. The slope of the line is lower here. It's as if the hippocampal memory trace lingers. The hippocampus is the memory trace is still hippocampal dependent for a longer period of time. It's as if consolidation doesn't fully occur. Remember, the new neurons have been killed by the X-rays. On the flip side, exercise has been shown to increase neurogenesis. And as more new neurons are born and integrate into the hippocampus, the effect of that is that the consolidation process is speeded up. So the memory seems to become hippocampal independent more quickly. It's as though the neurogenesis is triggering consolidation events and thus speeding up the independence of the memory. So the hippocampus is no longer needed to retrieve the memory. So we can think then of neurogenesis as possibly doing two things that seem contradictory, but if they're done efficiently and effectively, they go together. On the one hand, neurogenesis may be triggering system consolidations, so hippocampal memory traces are being used to strengthen cortical memory traces. But the very integration of the new neurons into the hippocampus is erasing the hippocampal memory traces. But the two can coexist. If the system consolidation is effective, then the hippocampus can tolerate the disruption caused by the new neurons. And so, in other words, the brain is kind of having its cake and eating it too. It's preserving the older memories up in the cortex and preparing the hippocampus for new learning. Again, the neurogenesis may help to preserve old memories by triggering system consolidation, but it's also preparing the hippocampus for new memories. So this gives us another reason for why the brain may be consolidating. System consolidation protects a memory from disruptive changes in the hippocampus. System consolidation allows the hippocampus to be maximally prepared for new learning without sacrificing older memories.